Welcome to our One Health Fish Monitoring Citizen Science Project findings to date. We hold these presentations every 6 months in efforts to try to provide updates to where we are on specific citizen science projects and where we're headed, so in which direction. And so this is our March 2024 findings to date. My name is Missy Weiss, Director of Citizen Science and Education at Orca, as well as the project coordinator for this particular project. And so just kind of assuming that there's no background to the project, I'm going to kind of just walk everybody through what the One Health Fish Monitoring Citizen Science Project is. Before I do that, let me talk about Orca. So the Ocean Research and Conservation Association, nonprofit, founded in 2005. Our mission is to protect and restore aquatic waterways and the species those waterways sustain. We do this in 4 major ways. We do this through our innovative technology, specifically our real-time water monitoring devices known as Kilroys. We actually have the largest real-time water monitoring network in the state of Florida at this time. And these devices are spread throughout the Indian River Lagoon and those critical freshwater canals to try to track pollution at its source. We also uphold this mission through our applied science research, where we focus on ecotoxicity and One Health, through our citizen science research, as well as our education, where we are able to talk to students of all ages, backgrounds, and introduce them to science research and applied science. Now, most of our efforts are focused in the Indian River Lagoon, given it's 40% of Florida's east coast and has tremendous ecological and economic value to our region. And so it's hard not to know that this waterway is experiencing lots of degradation, and it's being caused from lots of different issues. So habitat fragmentation, changes in hydrology, nutrient loading, there's so much, and it's incredibly complex and interconnected. And so one way that we try to overcome that complexity is by collecting as much information and data as we can. And so one way we can do that is with the help from the community through our citizen science research. So we currently have 4 citizen science projects. We have our habitat restoration work known as Land to Sea. We have pollution mapping, One Health Fish Monitoring, which is obviously the topic of today's conversation, and our A Day in the Life of the Indian River Lagoon. So before we really get into the data, I think it's really important that everybody understands what the term One Health means. So One Health is an idea or an approach that recognizes that the health of people is connected to the health of animals and our shared environment. And so when we protect one of these entities, we are indeed actually protecting all of those, because there's a clear interface where the 3 of these identities kind of come together. And so when the environment's health is impacted by contamination or pollutants, there's going to be some kind of health concern or concern to the animal and human health populations. And even the National Institute of Health recognizes that over 90% of human cancers are actually related to environmental exposures. And it's not just cancers, it's gastrointestinal disease, neurological disease, liver diseases, and more. And then also under the term One Health, there's this recognition that there needs to be efforts to inform and educate multiple sectors. And so this approach involves a lot of collaboration, communication, and coordination across different sectors, whether it's the public health sector, veterinary sector, or the science sectors. And that is all necessary in order to actually address the root causes of these diseases, promote health, and ultimately address the environmental concerns that are coming from all of this. And so where we really see ourselves at Orca is being able to collect the very needed data or evidence which is required to actually drive some form of change. And so for the One Health Fish Monitoring Citizen Science Project, the idea of this project is really trying to better understand, well, what are the pollutants that are making their way into the lagoon? And are those pollutants accumulating into fish that are commonly consumed by humans? and what could be the potential human health risk, if any, through the consumption of those fish. And so where we really see our data set going is being shared with those other sectors that were just mentioned. So being able to share our data sets with local human health clinicians, veterinarians, and people who can be more informed about, well, what are the toxins and toxicants entering in our waterway, and could they be impacting human health or patients? So this is really kind of the big picture of the One Health Fish Monitoring Project. At this time, we're looking for a panel of analytes, and we have been able to process or dissect over 1,100 fish over the last several years. So at this time, we are looking at microcystin, which is a biotoxin in our fish fillet. We're looking for heavy metals, primarily mercury at this time on our fish fillet, herbicides such as glyphosate, microplastics in both fillet as well as the stomachs of the fish. We have some new data tonight to share on forever chemicals or PFAS in fillet, and we are currently working with Embry-Riddle to develop the methods to analyze UV filters or the chemicals associated with sunscreens in fillets as well. And then crossing fingers, if we are to receive a grant that we submitted recently, we will also be expanding our panels of analyses to look at pharmaceuticals and controlled substances in our fish. So this is kind of the current panel. We at any time, whether we're working with other collaborators or we find new information or data or evidence of emergent concerns, of contaminants of concern, we can kind of go back and look at those pollutants on our fish samples. So to give you a little update on, see if I can press here. So a little update on the project itself over the last several years. This is definitely one of our more popular citizen science projects. We have over 250 volunteers. And I think it's because there's lots of ways that volunteers can get involved. So one way that you can get involved is by helping us collect angler surveys. So this entails going to the local boat ramps or popular fishing locations and interviewing individuals who are catching and consuming fish from our local waterways. That helps us determine what species we should be targeting, how often are individuals consuming these fish, how much are they consuming, and how are they sharing it? Are they sharing it with their pregnant wives? Are they sharing it with their elderly parents? It gives us that human health dimension that we need. We also invite people to participate by catching fish and donating the fish to the project, as well as coming into our lab. Usually there's an opportunity at least once a week where people can come in and help us process or dissect those fish, so remove the important tissues and organs that are analyzed. And then we also mentor and help other volunteers actually run the extractions. So they're extracting the toxins and toxicants from those fish samples. So there's lots of ways that people can get involved. And we have to, before I go too far, I just want to recognize and say thank you to our participating bait and tackle shops. So when people catch and donate their fish, they can drop them off at one of these local bait and tackle shops, which makes it really convenient. And we couldn't do it without them. So we always want to recognize and thank you for doing that. And also the Marine Discovery Center, which is not a bait and tackle shop, but they are our only Volusia County drop-off location. So we're very appreciative. And also when we're talking about what fish to donate, again, we're targeting fish that are specifically ones that have been identified through our surveys as being commonly consumed by humans. And so this is the list of saltwater and freshwater fish species that we are looking for people to donate to the project. They have to be legal, they have to be in season, and there's specific protocols that need to be followed. But this is really the list at this time, and it may change as more surveys come in that people are identifying as fish they're eating. So to jump into some of the preliminary results, I certainly can't go through all of the different analytes that we're seeing. So I usually just try to pick and choose a couple of them. to just give updates on. And so if you attended last year, then some of this may be familiar, but we've certainly added new data. And so let's start with how many fish we have coming from to each region. So we divide the lagoon up into a northern region, central region, and southern region. And so these are the fish that have been processed to date. So we've got about 450 fish from the northern region of the lagoon that encompasses Brevard County and Volusia County. We've got 638 fish from St. Lucie and Indian River County. And then Martin County represents the Southern Lagoon, and we just have 84. So we certainly need to bump up the number of samples we have coming from the Southern Lagoon. If we take this and we break it down further by county, you can see that most of our fish are coming from Brevard and Indian River County. We have a decent representation from St. Lucie County with 166 fish. And while Volusia, we have 8 lonely fish, that actually is an improvement since the last findings to date 6 months ago, because we only had 1 at that time. So we are slowly gaining fish from the northern region, and that's largely because of our partnership with Bethune-Cookman University. We have some wonderful interns who are helping collect surveys and fish from that area. And then we can even take it a step further, and we can kind of look at different sectors within each county. And so this is Brevard County. We divide each county up into little pockets or sectors, as they're called. And so each one of these colors is representing a different sector. And so BC 6, for example, is Brevard County, Sector 6. And we can start looking at fish from those specific sectors to see if we're seeing any changes in the amount of accumulation of toxins in those fish as well. So, we can really start targeting areas to see whether or not we're seeing different pollutants accumulating in fish. So if we start the conversation tonight with looking at one of our analytes, we're going to start with glyphosate. So glyphosate is an herbicide. It's the most commonly used herbicide worldwide. It is a billion-dollar business. But there is some contradictory information regarding its impacts on human health. And so according to the EPA, there is a summary review of glyphosate that states that it's unlikely to be a human carcinogen, whereas the World Health Organization says it's probably carcinogenic to humans. And so there's this contradiction between how it may impact human health. This is, by the way, the active ingredient in herbicides like Roundup. And so it's not only Roundup, there are other herbicides as well, but this is just one of them. But what we found through our own research here at Orca is that glyphosate is persistently found in our water samples as well as our sediment samples. And we know that there are known biological effects on different stages of fish development, impacting things like their behavior, their development, their reproductive abilities. And so there is this interest in determining whether or not glyphosate is accumulating in fish because we know it's in its environment. And so again, if we think back about that One Health term, we're thinking about, well, how is this pollutant entering into our environment? Is it impacting animal health, and could it be impacting human health as well? So we started looking at glyphosate in fish over a year ago. At this point in time, though, we have analyzed 393 fish fillets specifically for glyphosate. So we've increased our number quite substantially from the beginning. The way that we do that is we have people, volunteers, donate their catch to the project. Then we have other trained citizen scientists help us dissect those fish. We have other volunteers come into the lab and they homogenize those fish samples, which just means they grind it up so that it's uniform. And then they will aliquot 1 gram, where then we will have additional citizen scientists extract the glyphosate from that sample, and then Orca staff will run what's known as an ELISA, and that will generate the concentration. So of the 393 fish analyzed, 99% of them had glyphosate above a level of detection based off of the ELISA, which is 0.01 parts per billion or 0.05 parts per billion. The average glyphosate concentration across all fish fillets is just 0.25 parts per billion. So we're talking about very, very, very small concentrations. And it ranges from those individuals that are below the level of detection to the highest concentration of being 0.991 parts per billion, which was in a hardhead catfish. And so if we kind of, again, look at average glyphosate by region, again, Northern Lagoon, Central Lagoon, Southern Lagoon, we find that fish from the northern region are actually significantly higher in glyphosate concentrations compared to fish from the central and southern regions. Again, we recognize that we're only looking at a sample size of just 35 fish from Martin County. And so our sample sizes are quite different, but that is what we're finding at this point in time with the data regarding glyphosate. If we break it down further, again, looking at it by county, you can see that it starts out high in the northern part of the Indian River Lagoon in Volusia County, and then slowly decreases as you move your way south to the Indian River Lagoon. Again, just to kind of catch our bearings here, we've got county on the X. The parentheses represents the number of fish analyzed for glyphosate from that county. And then the y-axis is average glyphosate in parts per billion. What we're seeing at this point in time is that Brevard County fish have significantly higher concentrations in glyphosate compared to St. Lucie County and Martin County fish. So that is just has changed since the last findings to date as we've added more data. When we look at glyphosate, average glyphosate by species, again, we have a little bit of a different situation here. We have species on the Y, and we have parts per billion or average glyphosate on the X, going from lowest concentration to highest concentration. You don't see a species listed on this figure. It's only because we don't have at least 5 individual glyphosate tests for that species. We likely have data for that species, but we may not have at least 5. And so I really just wanted to kind of represent species where we had at least 5 individual data points. So from this figure, first and foremost, you can see that average glyphosate concentrations vary by species. Doesn't seem to correlate with size of those fish or diet. And there's no actual significant differences between average glyphosate by species either. But what I do want you to kind of recognize is that we're seeing the highest concentrations in species like black drum, spotted sea trout, our snook, red drum, and bluefish. And the highest concentration at this time is in our black drum, which is in 0.32 parts per billion. And that's going to be important for this next couple of slides. So again, remember, when we get this data, it's also about being able to try to understand its potential human health risk. And so when advisory levels exist, we often will compare our data to those. And so there is an advisory level, acceptable daily intake level for glyphosate in fish. And so if we just take that species, which is the black drum with the highest average glyphosate at this time, it's 0.32 parts per billion, then we can compare it to the current advisory levels. And so we'll do that now. So the first thing you need to do is you need to convert parts per billion into milligrams per kilogram. And so when we do that, we get this lovely number here, which is 0.00032 milligrams per kilogram. And then what we need to do is we need to understand, well, how much are typical individuals consuming? So if Joe here is eating black drum for dinner tonight, then what would be a typical serving size? So according to our surveys, that typical serving size is 8 ounces. And so if Joe eats 8 ounces of black drum, we know that based off of that average glyphosate concentration, he's consuming about 0.0007 milligrams of glyphosate for dinner. Now, like I said, there is an acceptable daily intake level. It actually depends where you live on what that level is. And so the United States' level is 1 milligram per kilogram of body weight per day. Australia and New Zealand, they're more conservative. Their value is 0.3 milligrams per kilogram of body weight per day. And so if Joe, we assume Joe is an average male, he weighs about 180 pounds. Again, we know how much in milligrams he's consuming. And so in order to reach that acceptable daily intake level, Joe would need to eat about 350,000 servings of black drum per day to reach that acceptable daily intake level, which is clearly not happening. And so at this time, we feel pretty good that the glyphosate concentrations in our fish, while they're present, and it certainly is an herbicide that is accumulating in fish, which is not good, it's not accumulating at levels of concern at this time. So that's a good thing. If we kind of shift gears a little bit and we talk about heavy metals and some of that data, we can see if that's the same case. So we have tested for fish for 3 different types of heavy metals. We looked for lead, cadmium, and mercury. And so just to kind of share 2 of them, the way that we get this data is, again, citizen scientists, after the fish is donated, they'll help us process it, they'll help grind it up or homogenize it, aliquot 5 grams into a vial, and then that vial is sent to a contract lab at Pace Analytics where it's analyzed for total mercury or cadmium or lead. So we tested 100 fish from an Indian River Lagoon small grant, where, let me put it this way, a small grant from the Indian River Lagoon National Estuary Program to test 100 fish for cadmium. Only 2 of those fish came back with detectable levels of cadmium. So again, once again, that's a really great thing. We're not seeing that cadmium staying in our environment and accumulating in our fish. We have continued to test for mercury, though, because we are finding that 99% of the fish that we test have mercury present. So we've tested over 1,100 fish for total mercury. The average mercury concentration in those fish fillets are about 0.2 milligrams per kilogram, and it ranges from below the level of detection to 1.8 milligrams per kilogram of total mercury. Now, what does that number actually mean? Well, before we get into that and how we compare it to human health advisory levels, it's probably important to just talk about how mercury gets into our environment. So a lot of mercury is going to be deposited into our Indian River Lagoon and waterways from atmospheric deposition. And in a lot of ways that mercury gets into our atmosphere is from combustion, industry, burning coal. And it's been determined that a lot of the mercury that's ending up in our lagoon isn't even from our area. It's being brought over with the winds and is from other parts of the world. So I'm not really sure what we can do to mitigate that, but it's certainly we can be aware of that. So if we look at average mercury per county, just as we did for glyphosate, you can see that it's not as, doesn't follow the same trend as how glyphosate accumulated in fish, but we're seeing a different pattern here. What we're really seeing at this time is that fish from Brevard County, and this is all fish, not just those that we have more than 5, but all fish from Brevard County have significantly higher concentrations of average mercury compared to Indian River County. But Martin County fish is really where we're seeing the highest concentrations overall, and that is significantly higher than all the other counties. And so this is something that we've seen even when we've had smaller sample sizes, is that on average, Martin County fish have significantly higher concentrations. Now, there are established mercury human health guidelines. They're established by the Florida Department of Health, the EPA, the FDA. And so what we're seeing is the same exact graph that I showed you prior, but now we have our 2 lines on top of that. So the orange line is the Florida Department of Health's advisory level for mercury for women of childbearing age and children at 0.1 milligrams per kilogram. And the green line is the joint advisory level for mercury for the general population. And so you can see that across the board for all counties, fish on average are exceeding the advisory level for women of childbearing age and children. But Martin County fish are exceeding the advisory level for the general population. So that is something that is certainly kind of perking our ears, and we are following. Now, again, remember, the intention of this is to be able to share this data set with decision makers, stakeholders, the community, so that they can make educated decisions on maybe what species they're consuming or how often they're consuming it, but also these agencies that are setting these guidelines. And so before we do that, we really want to bump this number up from Martin County to exceed at least 150 fish. And so we're actively pushing to acquire those fish also in Volusia County as well, but particularly Martin County because of these higher average mercury concentrations. So how does average mercury compare by species? This is, again, a list of some of the commonly consumed fish. In this case, we only utilized fish where we had at least 10 individual mercury concentrations per species. And so we have species on the X, we have mercury concentration on the Y, and this is the average of all of those mercury data points. So it's going from low to high. And so we're seeing that the highest average mercury concentrations are in fish like bluefish, crevalle jack, spotted sea trout, ladyfish, and hardhead catfish. When we kind of overlay those advisory guidelines that I just showed you for the county, we're seeing that there are a couple of species where that advisory level falls below it for the women of childbearing age and children. So Atlantic croaker and black drum appear to have average mercury concentrations that fall below that. But then clearly you can see that the other species listed here are exceeding that guideline for women of childbearing age and children. And there are actually 4 species, ladyfish, spotted sea trout, crevalle jack, and bluefish, that exceed the those joint advisory levels. And because that error bar of hardhead catfish exceed the green line, it means that in some cases, some of those particular specimens also did. So as we acquire more species, as we acquire more data, this bar graph will hopefully expand, and we'll be able to show how more data changes this, if at all, and how more species may be impacted. So, one of the things that we keep having conversations about internally is, well, are these mercury concentrations, if we're not burning coal, Florida doesn't burn coal anymore, if we're not really introducing more mercury into our environment, how do the current concentrations found in fish compare to historic values of mercury in fish? And so there isn't much data about fish in the Indian River Lagoon and their mercury concentrations. I was able to find 2 really. One is by Strawman Graves. It was done between the years of 1989 and 1999. So quite a while ago. And it was able to look at the species that are listed here in this table here. This is the number of each specimen they were able to acquire, and then the average mercury concentration. So for example, they tested 10 bluefish and found that the average mercury was 0.64 milligrams per kilogram. There was another study that I found from Adams et al. From only looking at spotted sea trout in the Indian River Lagoon in April and May of 2008. They basically tested 14 spotted sea trout and determined the average mercury concentration of 0.33. So even between 1989 and 1999, you can see that mercury really hasn't changed much in spotted sea trout. And so I wanted to be able to kind of overlay some of our data from our project and compare it to those historical values. And so I did this in the last findings to date, but again, we have many more samples and it's a little bit more representative. So this is the One Health Fish Monitoring Project. It's currently been done from 2019 to present day. And then it's the same format. So same species and the sample size in this column here, and then the average mercury concentration in this column here. And the final column is really, well, are we seeing any change between Strom and Graves study and the Adams study and our study. And so black drum wasn't ever tested, so we're kind of setting the stage for that. But it appears for a lot of these fish that we're seeing decreases in average mercury concentrations by the species. So for bluefish, crevalle jack, common snook, goff top-tail catfish, we're kind of seeing a decrease in average mercury, a slight decrease in mangrove snapper, but you can see our sample size is much different. A slight increase in pompano, but not really by much. In some cases, no change, but I guess this is a really great thing in that we're not really seeing increases of mercury. So I just wanted to kind of share this dig into some of the historical measurements of mercury in fish. And if we move to our final analyte that we want to discuss today, that is PFAS, or our forever chemicals. PFAS stands for polyfluoroalkyl substances, and they're also synonymous with the term forever chemicals because just that once they get into the environment, they stay in the environment, and they are quite persistent and pervasive. Now, this is new data. This was not presented last year, and I'm excited to share with you now. So PFAS is just kind of a catch-all for a series of compounds, and according to the EPA, there's over 15,000 PFAS variations. So there's lots of different compounds under this umbrella PFAS, and it's really being recognized as an emerging contaminant of concern. So there's lots of attention being focused on PFAS because, again, it is quite pervasive and persistent, and it's found in our environment, in our surface waters, in our groundwater, in our sediments. It's found in our wildlife, so our animal health, and it's found in our own bodies, so in our human health is being affected. And there's lots of sources of PFAS, which it makes this, contaminant of concern so challenging because it's really literally almost everywhere. It's found in our clothes. It's found in our non-cook cookware. It's in our personal care products. It's in pesticides, paints, fast food packaging, firefighting foam, firefighting clothing, photography. I mean, it is literally found almost everywhere. And there's a strong association with plastics as well. And it's been challenging because again, they're utilized for so many different products. Now, over 600 different species of wildlife have detected PFAS in that animal life. And so we wanted to be able to determine what's the PFAS concentrations in fish from the Indian River Lagoon, because there is known human health implications and animal health implications affecting things like cancers, cholesterol, child development, and so much more. And so there's been a lot of attention on and a big regulatory spotlight being shown down on PFAS contamination in drinking water, but there's not as much attention on PFAS in fish and how the consumption of that fish may be another mode of entering into human bodies. And understandably so that drinking water would be observed, but it's just another exposure route through our food sources. And so if we kind of focus on PFAS in fish, then the EPA knows that over the last decade, they've done testing on one of the most abundant PFAS compounds known as PFOS, and it was detected in 3/4 of all the freshwater fish that they analyzed. Because of this knowledge that PFAS is so prevalent in our environment is now accumulating into our fish tissue and therefore being transferred to humans, one being consumed, there's no federal regulatory on fish consumption for PFAS, but some states are starting to establish their own. So 17 states have published PFAS-related fish consumption advisories for at least one body of water in their state, but those levels vary quite widely. So if you can see here in the list I represent here, the Department of Conservation in Maryland sets a do not eat threshold of any fish that's at least 41 parts per billion. And that's just looking at that PFOS compound. That's not the most conservative state. New Hampshire at this point has the most conservative do not eat threshold at 25.7 parts per billion of all PFAS in their fish. But then you see here in this list that Alabama sets their threshold at 800 parts per billion of PFAS, or PFOS. So those advisory levels vary quite widely. In Florida, it doesn't even currently have a PFAS-related fish consumption advisory. So we're really hoping that the data from this project can be utilized to establish those guidelines. So we were fortunate enough to be able to receive another Indian River Lagoon National Estuary Program small grant, where we were able to send some of our homogenized fillet samples to our partners at the University of Florida, where they analyzed those samples for 31 different PFAS compounds. So we sent 111 fish of 10 different species. Again, those species were fish that are identified as being commonly consumed. That data has now come back, and the University of Florida detected 6 unique PFAS compounds in those 111 specimens. That 96% of those fish had PFAS detected, and not surprisingly, PFOS was most commonly detected in those fish species or specimens. And the highest concentration of that was 67.07 nanograms per kilogram. So when we compare that to those advisory levels that I just showed you, you can see that for Maryland, that would be high. And for New Hampshire, that would be high, but clearly not Alabama. Now, there doesn't seem to be any significant differences at this time between PFAS concentrations and county. This is just a breakdown of that, though. So fish from Brevard County, fish from Indian River, St. Lucie, and Martin County. We didn't have any Volusia County fish to send at that time. And this is the sum of all those PFAS. So, at this time, there's no significant difference between them, but we're seeing fish from Brevard County have the highest sums. And again, most of that PFAS is coming from PFOS. When we look at it by species, this is the 10 species that we were able to send, and this is the number of each specimen or a species that we did actually send to the University of Florida. So we sent 5 Atlantic croaker across the 4 counties to be analyzed, 7 bluefish, 8 common snook, 16 crevalle jack, and so on. So if we look at those species that had the highest overall sum of PFAS, we're seeing a lot of commonality between what we saw in mercury. So we're seeing higher PFAS in crevalle jack, hardhead catfish, ladyfish, and a little bit medium amounts in mangrove snapper. We didn't detect any PFAS in pompano, and minimal amounts in other fish species. Now, those fish, if we could look at certain species of fish that actually were exceeded that more conservative New Hampshire guideline, what we ended up seeing is that 8 out of the 111 fish actually were above that threshold set for New Hampshire. So if we were in New Hampshire right now, 8 of those 111 fish would be above that do not eat threshold. Those species were from crevalle jack, 1 mangrove snapper, and 2 ladyfish, and 1 hardhead catfish. So those were the 4 species that exceeded those levels. So kind of summarizing what we talked about so far this evening, what we're really seeing is that glyphosate in fish is really ubiquitous. It's found nearly in all fish, but at very low concentrations and at levels that are not of meeting that acceptable daily intake levels. We are finding that PFAS compounds are pervasive and accumulating in fish, just like they are in our environment. And we're seeing that mercury concentrations in fish vary geographically, with fish from Martin County have significantly higher average mercury concentrations compared to the other 4 counties, and that it varies in fish species as well. When we look ahead to the future and where this project is moving, we know that we want to continue targeting species and acquiring samples mainly from Martin and Volusia County, although we'll still accept them from the other counties. We want to dive into some of those freshwater fish. We have a lot of them, but we haven't really analyzed them separately. We want to start looking at relationships. So there's an interesting paper that just came out looking at that showed a strong relationship between microplastics and PFAS compounds. And so we're going to start looking at how our microplastics compare to that PFAS compounds. We're going to be adding additional analyses. So currently, as I mentioned, Embry-Riddle professors are developing the methods to look at those chemicals from sunscreen in fish. And so those methods are being developed, and hopefully over the summer, we'll have some students working on that. And then we really want to start looking at what is the cumulative impact of all of these pollutants on individual fish species, and how much would we potentially as humans be consuming when we consume those individual fish. And our ask of the community, well, get involved. One way you can get involved is, again, you can catch and donate your fish. They've got to be legal. They've got to be in season. You can come into our lab. You can learn how to process or homogenize our fish, or even extract them. You can participate in one of our upcoming fishing opportunities. So we actually have a day where we have one of our really active volunteers are going to head down to set up a table and tent and collect fish from that local boat ramp. And that'll be a great way to kind of get people in the community involved and knowing about the project, but also donating. So he'll be there from 7 a.m. to 4 p.m. collecting fish samples. If you live in Martin County, you can attend our fish monitoring intro and workshop, where again, we'll train you on how to process those fish. That's one of the few in Martin County that we offer just because of the distance. So if you know someone, definitely send them. We'd love to see you on May 14th. And then spread the word. So the more people that are talking about the project, the more we can disseminate information so that members of the community can make informed decisions and can be part of better understanding this One Health problem. And if you have any questions or you want to know more or get involved, you can always reach out to me. This is my email address. I welcome you to do so. And I just want to thank our citizen scientists who have been engaged in this project, donated so much time and effort, Orca staff and interns, our partner from University of Florida who's analyzed our PFAS samples, and then all of our sponsors who have donated the funding to allow us to do this incredible science, because science is not cheap and it takes a village. And so we want to thank everyone who has helped us and contributed to the progression of this project.